DNA replication. Objective. In this topic, we will learn about structure of DNA and replication process. The human body is made up of numerous eukaryotic cells. An internal view of a cell shows a centrally located nucleus. When we move inside the nucleus, we find the chromosomes. A zoomed-in view of a chromosome reveals the DNA. DNA shows a double-stranded structure, which is coiled in the form of a helix. One strand of the helix runs in the 5 to 3 direction, and the other one runs from 3 to 5 direction. DNA has the property of making a copy of itself, and this is known as DNA replication. At specific sites where the nucleotide sequence has repeated A and T base pairs is known as the replication origin. At this replication origin, the enzyme helicases gets attached to each strand of the uncoiled DNA and helps in unwinding the DNA strands. The hydrogen bonds between these bases are broken down. The unwound strands are further stabilized by several proteins called single-strand binding proteins, or SSB proteins. They prevent the DNA strands from coiling back. The uncoiled DNA forms a bubble-like structure called the replication bubble. Each of the uncoiled single-stranded DNA at the replication bubble then acts as the template for the replication. DNA replication always proceeds in the 5 to 3 direction. DNA-dependent RNA polymerase enzyme forms a small RNA primer on both the template strands at the replication origin. If we look at the template strand, which is oriented in 3 to 5 direction, an enzyme called DNA polymerase gets attached to DNA near the primer. Another molecule of this enzyme also progresses in the 5 to 3 direction. Meanwhile, extra nucleotides approach the replication origin. The DNA polymerase starts joining the appropriate complementary nucleotides to the template strand. In this case, the replication is continuous, and thus the newly synthesized strand is known as the leading strand. If we shift our attention to the other template strand, the movement of DNA polymerase on the RNA primers occurs from the opposite side. Thus, after addition of a few nucleotides to the primers, replication stops. Thus, several fragments are formed. These fragments are known as Okazaki fragments. Replication process on this strand is discontinuous. Thus, the newly synthesized strand is known as lagging strand. Another DNA polymerase enzyme replaces the RNA primers with DNA molecules. DNA ligase enzyme seals the gaps in the lagging strand. Eventually, 2 daughter DNA strands are produced. After the replication concludes, the uncoiled DNA starts recoiling and forms a double helix. Thus, 2 DNA helices are formed from a single helix.